Hi all. Forgive the fact that you have me and not Natalie leading this today. She had to excuse herself, so I'm jumping in. Let's see, make sure everybody's muted as we do this. And here we go. Okay, so welcome and thank you all for coming to an sort of off-hour CBCS seminar. We normally do this an hour later, so it's great to see everybody here. And I would like to welcome Cynthia Riginos and Maria Bager. It's lovely to have them, and it's lovely to have Maria back. She predates me by a long shot in CBCS, so it's wonderful to have members of the family come back and present on some of what they're doing. Today, they're going to talk about how can evolutionary genetics contribute to spatial planning, and I will let them introduce themselves as they get started with the presentation. But thanks again, all, and thank you to Maria and Cynthia for contributing here. Looking forward to it.
Great, thanks, Daniel. And I wanted to thank Kate for organizing everything flawlessly as usual, and for Maria for making the valiant effort to stay awake, which Maria's in the UK right now, and so we shouldn't ask too much of her. So I'm just gonna, this talk is, I haven't done this talk before, so I think it really will only be 25 minutes to half an hour in length, but I'm totally happy for people to jump in at any point. Like, I view this more as a discussion and things that Maria and I have been thinking about for a long time, and so we're really interested in other points of view. So Maria can correct me if I get anything wrong, but other people, if you have questions, clarifications, comments, and you want to jump in midstream, that's totally okay with me. I do a lot of teaching, so I'm pretty used to that sort of style of talking. So Maria and I just finished an epic journey of writing a chapter for a book in a very short period of time, which I feel that we're a little bit bruised about. And it is nominally on corals, and that will be coming out in the next year. Now, that chapter sort of gave us an excuse to consolidate a lot of things that we've been thinking about for a period of time. So we're hoping that some of the ideas that we kind of gave a test run to in a very coral-centric framework are something that we can gain traction for in a non-ecosystem, non-species specific sort of way. So we're really trying to think about generalities, and this is why I thought it'd be really nice to talk to this group to get comments and feedbacks and questions from people. So a lot of the examples I'm going to give are corals, but we're not necessarily just thinking about corals today. So you know, I guess I'm a newcomer to CBCS, which is a little bit weird, but anyways, we won't go there. But I thought I'd say a few things about who I am, if some of you don't know me. So my group works on a whole variety of different organisms. We really can't focus on anything in particular, it seems like. They're mostly marine organisms, so we mostly work in marine systems, and we cover a variety of different topics, and some of these things over here that sort of loosely fall within evolutionary genetics and molecular ecology type of framework. So I particularly wanted to highlight conservation genetics and just kind of make an aside comment on conservation genetics, which is, I think when most people think about conservation genetics, they think about using genetic insights to help increase the likelihood of continued survival for species that are already imperiled, and tends to be focused on a particular species. And that's fine, I don't have any problems with that, but that's not what I do. So the sort of conservation genetics that I do is a little bit more abstract, in the sense that I like to think about fundamental questions in biology, try to understand how fundamental processes work and extract generalities, with the aim of those generalities being something that can have implications for conservation. So much more abstract, but at the same time, I'd like to think about multiple species, about communities and so forth, not just focusing on one particular species. So that's a little bit of spin on conservation. So then, of course, my partner in crime, who should be asleep, but she's not asleep, she's bravely up, is Maria. And many of you will know Maria from her long-standing associations with UQ that she still maintains. And Maria is a dear friend of mine and a valued colleague, and it's really great to have somebody to work with. So Maria and I have been arguing, chatting, discussing some of the issues that I'm going to talk about today for, I think, almost 10 years. And that's really good because, as some of you will know, doing interdisciplinary collaboration is really, really hard. I think you have to maintain a really strong friendship with your colleagues because you have to have the comfort level to ask stupid questions and to get out of your own headspace and to try to understand what a different field's point of view is. And it's really, really hard. I think it's really exciting. And sorry, I turned off my notifications, but they seem to pop up. Technology. Anyway, so collaboration is really hard, but I think it's really fruitful as well. So once you find somebody in a different field to yours that you can work with, stick with them because it's that's the way forward, is to have those sort of associations. So we're in a present day where things are massively messed up, right? Humans are doing all sorts of horrible things to the environment, and I think all of us are acutely aware that the next couple hundred years are going to be really bad for a lot of organisms on Earth and a lot of species. And so in response to climate change and other anthropogenic perturbations, we see phrasing such as this, that the strategies that species are going to have to follow are either one of movements or range shifting adaptation. So that is evolutionary change to different circumstances, moving into different niches is another way to look at that, or they might die, which is of course what we would want to avoid. So from my point of view, somebody who comes from an evolutionary biology background is very well suited to make comments on these sorts of processes because these are the processes that many of us in that field study. So things like dispersal, natural selection, that's kind of my bread and butter in terms of what we study. I try to avoid extinction, but certainly there are many other evolutionary biologists who try to think about extinction. So we don't really want extinction to be happening. And we'd like to think about the circumstances that we can create for species that will increase their likelihood of being able to move or adapt to changing circumstances. So that's a kind of the coming in from the evolutionary side. But then what are the data that spatial planners use? And this is where I'm going to get way out of my comfort zone and probably say something that's completely wrong. But Maria made this figure, and so I'm going to blame her if anything goes wrong here at this point. So this matches my understanding what sort of data that spatial planners use and what sort of objectives that you might have. So you have all these goals here on the left, all these things that you want to do in order to come up with an overall plan. You might have these different factors that you want to take into consideration. And in order to operationalize this, you need to translate this into a spatial representation of some sort, right? So you're going to have a focal area, and you might also be interested in habitat. And then what I'm going to focus on today are these aspects here, which is how can we incorporate information on dispersal or adaptive potential. And I'm actually going to start with adaptive potential and then talk about dispersal, largely because for a long time I felt like adaptation was a harder thing to manage, but now I've actually convinced myself that we can probably do a better job incorporating adaptive potential in planning than dispersal. So interested in some comments there. So can we consider adaptive capacity or potential as a conservation feature? And I guess for me, I always like definitions because I think, especially when we're in the interdisciplinary space, you can't assume that your definition is the same definition that somebody else is going to bring to the conversation. So, what are we really talking about when we're talking about adaptive capacity or potential? So what is it, right? So one point of view is it's a specific trait or attribute. So with corals, people are really interested in increased thermal tolerance. So, corals that can withstand higher temperatures is a trait that is viewed as probably adaptive in the present and near future. But we can also think about it in not as tied to a specific trait, because a specific trait presumes that we know what's going to be important in the future. And so I guess I would come up with an argument that we probably want to think about just sort of the overall potential for evolutionary change, because we don't know what the future selective forces might look like. And that in some ways is a harder thing, but actually maybe it's an easier beast to tackle. So I think these are our main, when we're talking about adaptive capacity, this is loosely what we're aiming towards, but how do we go about measuring this? So this is, I'm going to give you a completely gross simplification about how evolutionary geneticists measure these type of attributes, and those geneticists who are in the room, I'm really happy to talk to you later about the details, but I think everybody else would fall asleep. So warning, this is like a gross simplification. So if you're interested in a specific trait or attribute and understanding what is the genetic basis of that trait, so that you can go out into nature and figure out what sort of variation there is in different locations within different species, you're probably going to be turning to quantitative genetics, which is an entire disciplinary field that involves setting up breeding experiments, measuring traits in multiple generations, perhaps intertracking that with genetic markers as well. It sort of depends on exactly what you're doing. But at the end of the day, regardless of what particular experimental design you're following, is that it's quite laborious. And really, people typically will create one or two family lines. So it's representative of a single species from a single place at a single point of time, raised under one environment. Okay, so you can already start to see how that might be problematic in terms of trying to think in the bigger picture. So that can uncover what are the parts of a genome that are important for a particular trait, with a lot of work and a lot of caveats. Another approach that people are turning to a lot, and particularly in population genomics and landscape genomics, is doing this thing called outlier tests and various tests of selection, where basically you get a lot of genetic information from a lot of individuals within a species from a lot of places, and you look for kind of your outlier loci in terms of loci that show sort of strange patterns of variation relative to the sort of the mean or the normal pattern. And you sort of say, okay, well, these are going to be under selection. And so those are the loci I'm going to be interested in. And again, I'm happy to talk details with people if you're interested in it. But it's really not clear how the loci that are being identified affect traits. There's a lot of spurious associations that can arise from this sort of program. And again, it's pretty much limited to one or a few species. So we can sort of scale up in space from quantitative genetics, but really you're not going to be doing this for lots and lots of species. So there's a little bit of a scaling issue as I see it. Okay, so what about if we wanted to think about the potential for evolutionary change to many unknowable selective forces? In some ways, that's a worse problem, but in some ways, it's an easier problem, because we can just say, right, we're just going to ignore a lot of things, right? So not clear what traits to measure. So maybe we just shouldn't measure traits at all. And there have been a number of really compelling arguments recently that we should just look at total genomic variation. This is a little bit of a debate in the literature, but I think there's some really good cases to be made, and these two papers at the bottom, if you're interested in this topic, are good starting places. The good news is that total genomic diversity is imminently measurable. We can do this. It's rather straightforward. And better yet, if we're interested in total genomic diversity, then we can subsample a genome, right? So standard sampling techniques, which means we actually don't need genomic level data necessarily, or we can get a reduced snapshot of a genome, which makes the gathering of empirical information much more tractable than some of the other methods. It's still a big ask, ultimately, and you're probably going to be limited to a couple of species at a time in a landscape, but it is much more tractable than some of the other ways that we could measure adaptive capacity. So I personally think that molecular ecologists can provide this information for species. So like if we have a hypothetical coral, we could tell you what does the adaptive capacity look like at these different locations based on measures such as total genomic diversity. Okay, of course, the spatial resolution is going to be less than would be desirable, but there's sort of some methods that we can deal with that sort of issue. So I'm sort of provisionally putting a green tick against adaptive potential. I think I think it's still a big research program, but it's something that is feasible. We can do it, okay, with caveats like usual. So what about dispersal connectivity? And this is actually where I think it's a harder problem the more I think about it, and the more that Maria and I argue about things and discuss things and ask each other dumb questions. So first of all, what are we really talking about when we're talking about connectivity? So it's useful to conceptualize connectivity as sort of a continuum, where at one end you might have demographic connectivity. So this is large numbers of individuals moving between different locations. The standard definition is that it's sufficient numbers of individuals that can affect population size. So think about, like in a metapopulation, if you have a location that's wiped out, you have an influx of immigrants and you can restart that population, for example. That would be strong demographic connectivity. At the other extreme, we have evolutionary connectivity, and this is sufficient to be evolutionary significant, which means it affects gene frequencies. It does not mean you have a lot of individuals. The rule of thumb is one migrant per generation is enough to spread allele frequencies around between locations. Now you can, if you're an ecologist, one migrant per generation certainly doesn't sound like very much. And it's certainly not going to repopulate a population or affect the population size in any measurable way. So I think we have a sort of a field disconnect here, in that planners are probably much more interested in demographic connectivity, whereas geneticists are much better at measuring evolutionary connectivity. So, how are some ways that we can resolve this mismatch problem? And so one of the things that Maria and I have been doing is thinking about what are the spectrum of methods that are available to us, and kind of exploring some of the different tools that might be out there. And so I'm sort of loosely organizing them on a scale of demographic to evolutionary connectivities. And in this sort of abstracted view of dispersal, this is a dispersal kernel, if you haven't seen this before, it's just a probability distribution of dispersal in a kind of spatially agnostic setting. So we're sort of ignoring all the things that might happen in a real landscape, and just sort of coming up with an abstracted view of dispersal. And what I've done is mapped on some of the methods that we can use using genetics to estimate, and where they kind of fall on that spectrum. And I'm going to lump these two together just for our quick conversation. So I'm just going to give you an idea of what some of these methods look like, again, in the sort of loosest way possible. So if you have any questions, I'm happy to chat further in question time or at lunch. So the easiest one conceptually to understand is measuring parent-offspring distance. So the basic program is you pick some focal locations, you work up the genetics on all your individuals in a focal location, And maybe, if you're lucky, if you've sampled in the right way, you'll be able to identify some babies that have parents that are within your focal area, and you can literally measure the distance between them. So clearly, there are going to be some issues and biases with this sort of a method, in the sense that, like these babies over here, we don't know where their parents are. We don't know who their parents are. So parents could be, that could be a parent, that could be a parent, or the parent could be, like 10 kilometers away, but we don't know. So that's going to be a problem. On the other hand, this method's really, really satisfying. Like when you identify these pairs, like that's cool, right? I think it's really neat when you can find parents and offspring. So that gives you some idea on really small scale dispersal, because what is the size of a transect or a quadrat that you can set up and exhaustively sample individuals? That's not going to be very big for most organisms. Another set of methods basically uses patterns of spatial autocorrelation, and these are very similar to each other, which is why I'm putting them on the same screen. Here on the left, we're looking at relatedness. That's like similarity. It's not literally relatedness necessarily, but this is on an individual basis. So the idea is that close together, you have higher related individuals, and as if you move further and further apart, if there's more dispersal happening, your individuals are on average going to be less related to each other. So that's looking at individual genotypes. It's just a spatial autocorrelation pattern. The other methodology is focused on populations, and we tend to look at differentiation. It's just a field, one of those field reasons. I mean, you could flip it around, it doesn't really matter. But the same sort of logic, like things that are closer together are going to have less genetic differences compared to populations that are far apart that are going to be different in allele frequencies. Okay, and so we can take lots of pairs of individuals, or we can take lots of pairs of populations, and we can come up with pairwise metrics, and we can fit regression lines to them. And if we also have information on population abundance, these slopes can be transformed to provide an estimate of the average dispersal distances. Okay, so this is again sort of spatially agnostic. It's a kind of over generic landscape, but we can come up with the average distance between a parent and an offspring in using these methods. So I think these methods actually have a lot of promise, and they're underutilized right now, in the sense that this is relatively easy to do for a decent number of species. It's not hugely involved in terms of the molecular side of the investigation. And then finally, we sort of have classic evolutionary methods that are probably not very relevant to most of you, which basically requires genomic data and is concerned with estimating gene flow over really large spatial distances, because it really estimates very low levels of gene flow. On the positive, these methods are pretty good at measuring asymmetric gene flow. So you're getting much more directionality information out of this, whereas this previous method that I was talking about has absolutely no directionality in it inherently. So there's evolutionary methods, but they're probably not very useful for spatial planning, because we're talking about pretty low levels of gene flow in order to be measurable during using these ways. So we've been focused on measuring connectivity, and that's been kind of our emphasis, like how do we put a valuation against that? But I think we might want to think about flipping this problem around and focus, rather than focusing on connectivity, maybe we should be thinking about isolation. And I know this sounds like the same thing, but not 100%. And in particular, one of the things I just want to note is geneticists are very good at detecting isolation. So if a population geneticist says, hey, these two groups are not interbreeding with each other, you can be pretty sure that they're demographically completely not connected to each other. And so maybe we should be concentrating on that, because that's something we can measure with confidence. So how do we go about doing this? Just to give you a taste of how this methodology works, we genotype a lot of different individuals, and then we use a k-means clustering type of approach. And then this depiction is indicating an individual's genotype. So like this individual on the far right would have basically 100% identity to the dark green cluster, whatever that is, whereas this individual on the far left is mostly associated with the light green cluster, okay? And this sort of pattern, you see this really commonly. You might have multiple individuals represented, as a population, okay? And so what's really nice about this sort of method is this captures isolation that might be very recent. So only a few generations can be captured using these methods. And so I think that aligns quite nicely to the time scale that is relevant for spatial planning. And we can take these outcomes and plop them down in a map, so you can have estimates of your individual assignment to different groups in a spatial manner, in which case, like in this hypothetical example, we see mostly dark green on one part of our landscape and mostly light green on another part of the landscape. And so we can conclude that these things are really not well connected through here. So there might be a lot of connection within these different regions, but there is no connection happening between these regions. And so I think that's information that could possibly be used in thinking about spatial planning. How you'd implement that, I don't know. That's not my field. I'm just gonna hand that over. So I think molecular ecologists can contribute to our empirical understandings of dispersal, but the genetic parameters will probably need further processing and modeling before they're at the stage where they can be useful for planners. So, this nice graph that is put down here, maybe we could come up with some of those values against some of those arrows in some circumstances, but most likely we're going to have to be fitting various biophysical models of dispersal against our raw estimates of dispersal that come from genetics. And so I feel like there's a lot of stuff that could probably happen here. And these are some nice papers that have tried to tackle some of these issues that were published recently and have people on them that you might recognize. So I guess in terms of thinking about dispersal connectivity, I'm going to put a very pale tick against that, because I think we're really not there yet in any way. I think there's potential, but we really need to engage a lot of really smart people to think out some of the ways that we can actually take the sort of information that you can glean from genetics and port it into a framework that is useful in terms of thinking about spatial planning. So just to kind of wrap up, some of the challenges that Maria and I see, and there are many challenges, but I guess these are some of the main ones that we see that require action and thought in the short term, is that there's a mismatch in scale and resolution. So generally we get empirical data for only a few sites, whereas planning you need to consider a whole area. So clearly interpolation is going to be useful, and dispersal modeling is probably necessary. So it's not a straightforward connection between the information and the outcome. I think another really important issue is that in some ecosystems, asymmetric dispersal is likely to be important. This is particularly so for marine systems that I study and Maria also studies. For genetics, we don't really have good methods for empirically estimating demographic connections and giving you asymmetric values on that. We just don't have that tool set, and I don't even know if it's feasible, honestly. I guess from the planning side, directionality is not well handled in present methods either. That's what Maria tells me. So, go ask Maria. Also, we're not very good at measuring and incorporating variance and uncertainty. So from the genetic side, many of our metrics are time average, but they don't give us the variance over time. And so that could be important depending on your goals. And similarly, from the planning side, those things are not really well managed in models. I think another really important aspect is we need to be planning for multiple species. So, we're really interested in whole ecosystems and communities. And so that is a much, much bigger challenge, right? So from genetics, we rarely have empirical data for multiple species. From the planning side, the theory and methods do not accommodate connectivity across species in a manner that's biologically meaningful. So I sent my slides to Maria just so she could have a quick comment, and she sent me back this huge, lovely email, which there's no way I can paraphrase it. So I'm just going to let you read Maria's response for a minute. So clearly there's some thinking there to be done, and maybe we can talk about that in the question time. So finally, thinking about a future world, our past conditions and priorities might not be relevant. So dispersal or adaptation that happened in the past might not be relevant for the future. So again, I think there's a real role for modeling of some sort and simulations. How we know whether it's on course, that's a big question, right? So I'd be really interested in other major challenges that people in this space. Maria, did you have something to say? No, you turned on your video. So I think both of us are really keen to hear about other challenges that people in this room who are very smart come up with. So I think clearly we need to be able to scale up. And as I sort of laid out in the beginning, the way I see a role of genetics is to help to understand some general principles, but we're not going to be able to go out, and much as I would like to go out and look at the genetics of lots and lots of different species, we're really not going to be able to do that at a scale that matches the information we probably need. So we need to start thinking about surrogate measures so that we can scale up. And I guess I see this as a research challenge for evolutionary geneticists, that we need to make sure that these measures, that these possible surrogates are actually accurate and actually measuring what we want them to measure rather than jumping in and assuming that they are. So in thinking in this space, some of our candidates for adaptive potential are looking at genetic diversity overall, which I talked about earlier in this talk, which we could use genome-wide diversity or a subset of the genome. But also there's some argument for local heterogeneity. So if you have a population that lives in a very heterogeneous environment, probably you're going to have individuals that have the genetic makeup that enable them to match that those different attributes of the local environments. And so within a heterogeneous and local area, you probably have pretty high genetic diversity. And so that might be something that we can use as a signpost for adaptive potential. We could also look at populations that have experienced future stressors, right? So if we know what's happening, I mean, we know things are getting warmer, where are the locations that have already experienced that and are still hanging on, because they might have the variation that we want in the future. And the final one that might not have occurred to some of you is that hybridization could be something we want to think about in prioritization, because there's more and more evidence that low levels of hybridization actually is really important for adaptation. So we as humans, we have some alleles that come from Neanderthals, and depending which where your ethnicity and ancestry is from, you might have some Denisovian alleles as well. So we have benefited from introgression from beyond our species. And so that might be something that we want to think about for other organisms as well. very interested in other ideas for adaptation surrogates that people might have. And then thinking about connectivity, some ideas that we had in terms of connectivity, this habitat configuration, that's kind of a stock standard one, right? That, you know, good patches that are close together are probably going to be more connected. Large patches are going to produce more propagules that can move between locations. Kind of a no-brainer, but we really need to know whether that actually holds together in terms of real connectivity. I think we're going to have to be using dispersal models to get at connectivity. There's just no way around it. And I'm interested in any other ideas that you might have as well. So as we know, the world is changing, things are bad, and I'm hopeful that all of us here in some very small way are going to make contributions towards organisms having a better chance of moving or adapting and not dying. I think that's the goal that we all share. And just to wrap up, I wanted to say that it's great to work with dear friends like Maria. This is the last time Maria and I were in the same place back in 2020 when we could travel the world. It was the last time I left Queensland. It seems like an eternity ago, with some other fine people that we collaborate with. So I think we're all looking forward to being back in single places where we can have some of those really detailed conversations. So thank you.